everybody. It is October 18th, 2024, which means we are 1 week away from the International Summit on Hypertrophic Cardiomyopathy and a little more than a week away from the actual annual HCMA patient meeting in Boston. There are tickets still available for the patient meeting. The summit has sold out in person, but I'm glad to tell you that there is a virtual option for those physicians who wish to attend virtually. You can still do that. So we'll be dropping some links here during the podcast to make sure that you know how to register for both the HCMA annual meeting and for the International Summit. Now that I've gotten my announcements out of the way and you know what day it is, you always know that here on Tales from the Heart, I announce the day of the month, year, everything, so you understand where you're hearing this technology information and updates from. If you're listening to this in a year or two, the data may have changed, so you may want to come back and look at us again. But today I am really excited to have a great conversation with Whit, who is coming to us from Tenaya Therapeutics to talk about genetic therapies in hypertrophic cardiomyopathy. And Tenaya Therapeutics dropped a press release yesterday with some exciting updates, and we're going to dive into all things genetic therapy. I default to genetic testing because we've been talking about that for so long, but we're going to talk about genetic therapies. Welcome to Tales from the Heart, and could you please introduce yourself to our audience?
I'm a research cardiologist. I studied at Hopkins, got both MD and PhD degrees, and then did my medical residency there. I came to California, where I'm calling from now, to get cardiology specialty training at the University of California, San Francisco. And I joined the faculty cardiology practice there while doing my postdoctoral research training.
And what was your postdoc in?
My postdoc was in arrhythmias and the mechanism of arrhythmias, and actually screening for novel genes that could contribute to making arrhythmias worse. And I took a career turn at that point because there was a company working on diagnostics looking at some of those same genes that I was looking at. So that was almost 17 years ago now, and I really haven't looked back. I've enjoyed working in biotech. I spend every day thinking about how we can help meet patients' needs, leveraging new science to develop better drugs. That's what I've been doing for the last 17 years. I've been at Tenaya for almost 6 years now. Before that, I was at Cytokinetics, and I had the pleasure of working with that team on aficamten, and I've been excited by the progress of aficamten recently. Tenaya, I'm the chief medical officer. We focus 100% on heart disease, and our goal is to address the underlying causes of heart disease. We have a particular focus in genetic cardiomyopathies and efforts to correct the genetic changes that cause those diseases. So we have 2 programs in clinical trials now, one for arrhythmogenic right ventricular cardiomyopathy, or ARVC, and of course one for hypertrophic cardiomyopathy, which we'll focus on today.
We will. And in full disclosure, the gene that you're targeting is the gene that runs in my family. That is not by design, it's by coincidence. And the myosin binding protein C mutation is arguably one of the most common. It's in arrays for myosin heavy chain, MYH7. But today we're going to be focusing on that MYBPC conversation, and super excited to hear what's going on for our friends in the ARVC community. But today we're focusing on HCM. Can you tell our listeners, what does your title mean? What do you do? What are you responsible for within the organization?
I like to say, I oversee science for patients. And so my work starts coordinating with the research team. We have an amazing set of scientists. I'm not at the bench doing the science. I can't, do what they do, but help focus what experiments should be done in the direction of what is most likely to move the needle for patients ultimately. That's why I love my job so much. Science, you do experiments, always raises new questions. But when you choose what question to answer next based on what could help a patient most, that's what gets me up in the morning. And then, so identifying, new targets, new molecules that drugs could correct in disease. And then how to get it safely into clinical trials with patients. So all the preclinical work that we do and discuss with regulators at the FDA to allow us to enter clinical trials. and then designing, conducting, and reporting the results of clinical trials to move the drug to the point first where we know it's working, and next where the FDA and the community know it's working. And at that point, we submit for drug approvals, and then it can be used in clinical practice.
So today we're going to be talking about a clinical trial in gene therapy. And gene therapy is a pretty new concept to most people, especially in cardiology. And I want to take a few minutes to kind of set the stage because terms matter. And there's a difference between genetic therapy and genetic editing. Could you start us off with a base understanding of what the difference between editing and therapy is?
Yeah, there are different types of genetic therapies, and you have to choose the type you need based on the way the gene is causing disease. So we're talking today about a gene replacement therapy. So folks like you and the members of your family get hypertrophic cardiomyopathy because that gene isn't producing protein. It's just not functional. And so we just need to add another copy, a correct copy of the gene to the cells in your heart. So that's gene replacement. And that is what our drug candidate TN-201 does. Other genetic variants cause disease by creating mistaken proteins, poison proteins that are produced, but they're disrupting the cell. So you actually have to get rid of bad protein as opposed to replacing protein. And so editing is one way to do that. And editing actually goes and corrects the misspelling in the DNA that's creating that dysfunctional bad protein.
Now, there are other trials going on in genetic therapies in the global sense. How is what you're doing different than editing?
I would say that as you say, gene therapy is relatively new in the cardiology space, but there have been tremendous successes in a variety of genetic diseases affecting many organs. And it's only very recently we've had the tools to deliver genes to the heart. But there have been 7 approvals for gene replacement type therapies similar to ours for diseases of the eye, the brain, the liver, and skeletal muscle, with approvals both in the US by the FDA and in Europe by the EMA.
It's really an exciting time for this concept. And I was like kind of looking back in history and figuring out how we got to where we are. We went on this quest in the United States to map the genome and figure out what this thing is doing. And in all of that work, we were very focused on discovery, but now we have the opportunity for action. And it's really an exciting time. It's a little, next level, if you will. And I think people are going to have to get used to some new terminology and words and concepts to understand how gene therapy may be right for them or their family members. And having conversations with you and others in this format hopes to kind of open up the door a little bit to some good conversations. So as we are going through this, if you have a question about genetic therapies, please put it in the chat area of whatever streaming you're seeing us on, LinkedIn, Facebook, YouTube. Let us know what you're thinking, and we're going to try to get to those questions at the end of this particular session. We know that gene therapy has to come a long way to prove its concepts. So can you tell us a little bit about how we're evaluating, how you've built this research protocol, and what's involved in the protocol, and what are the hopes? It's a lot in a question. So let's just talk about the trial up front, and then we'll kind of talk about the hopes for what we're going to develop. So what does a trial do? How does somebody find out about it other than this podcast?
Well, you can go to our website, hcmstudies.com, HCM for hypertrophic cardiomyopathy studies.com. There's a terrific government website, clinicaltrials.gov. I know some people find it hard to use, but it is really a great compendium of all the available trials out there. So I encourage people to learn how to use that. But I think our website is a little bit simpler and more focused on our genetic therapy trials. And we're, of course, always available for direct questions. Our email is patient.advocacy@tenayathera.com. So many ways to find us. The primary goal of this first in human trial of TN-201 is safety, to ensure that we are bringing this drug to patients safely and to measure safety as we go in the trial. So this is a one-time dose. So it just takes one intravenous infusion to get the delivery mechanism to deliver the normal copy of the MyBPC3 gene to the heart cells, and then it stays there and expresses the protein correctly from there on out. We will be monitoring closely how the body reacts to that initial infusion, and then how it adapts once the gene expression is restored. We know from preclinical studies in animal models that we can stop the progression of disease and even reverse elements of the disease in the models, including reduction of the hypertrophy and improvement in survival. Of course, these are animal models, and it's, people are not.
They're not men are not mice.
But that is our vision, is to achieve those same dramatic effects in people by correcting the underlying genetic cause of the disease.
So I'm going to break this down into some concepts. You tell me if I'm right or wrong. So there's this capsid that has a protein in it. That protein in the capsid is put into the body via an IV. That capsid finds its way to the heart muscle because it's programmed to find the heart muscle. It gets into the heart muscle and it delivers a patch, a fix, correct material for the heart muscle to pump properly. And that sounds pretty easy in concept, but it's highly specific and it is highly focused on getting to the heart muscle. Now, there's been another trial in an HCM spectrum disorder called Dannon's disease, in which they tried to get an item similar delivered to the Dannon heart to make correction there. Can we talk a little bit about what we've learned from the Dannon study and how it might guide us in the HCM world?
I think you described it very well. I will make a small adjustment. What we're delivering to the cell is the instructions for how to make the protein. The cell has all the parts for the protein, the pieces, it just needs the instructions to put it together. So we deliver the instructions, the cell puts it in its instruction manual, which is the nucleus with the genome, and then it can continuously make MyBPC3 protein correctly after that. We are delivering the instructions, the same tools as are used in that Dannon study. And we are happy to learn that that program is in phase 3, and then that phase 3 study is fully enrolled, which means that when the results come out of that study, the FDA can look at that and determine whether it's safe and effective for approval for general use for Dannon patients. They have come very, very far. What we know, what's been described about their first in human study, which was the study that supported the phase 3 study, was they found an appropriate dose that is appropriately tolerated and successfully delivers that set of instructions, in this case for a protein called LAMP2. And that protein is made in response. And we have data out to 3 years. The heart is continuing to make the protein at least 3 years after the one-time dose. And the way those instructions get into the instruction manual and the nucleus, we think they can last for many years. So it's not surprising that it's still working at 3 years, and we're hoping for many more years for those patients.
That is really encouraging. I've been working with Rocket on the Dannon's project for probably 5 years now. And it's been really exciting to see how genetic therapies are evolving, how the clinical trial environment is meeting the needs and scaling up to deal with a trial that's different than a drug trial. So we've been doing a lot of work to try to get people ready for the concept, and it's happening. It's happening right now. So nearly a year ago to the day, we're like within 24 hours of the anniversary, the very first patient in the United States was dosed with your molecule or your therapy. And it's been a year. I know a lot of people are asking me what happened. And I was excited yesterday to learn that in December, we're going to get some data. So we're not quite there yet, but something else happened yesterday. You guys dropped this press release, which tells me as a patient advocate, there's some positive signals here because your data safety monitoring board has made some changes, and data safety monitoring boards don't make changes to be less conservative if there's not something to do that about. So we can look at the inference here. And can you tell us what the data safety monitoring board has done and why that's significant?
Yes. So I described the primary objective of the study is safety. Another objective is to find the best dose. And we are excited to announce that our DSMB has agreed that it is appropriate for us to test the next dose level. So we have gone to a higher dose. We don't know which dose will be best, but the safety and tolerability we've seen so far is appropriate, and appropriate even for escalating the dose. But we are now enrolling patients at that new higher dose.
So,
in addition, as you alluded to, we are expanding the study based on the data that we have now. So we've increased the number of patients that could be enrolled in the study and broadened the eligibility criteria. So initially we were studying specifically people with MYBPC3 and non-obstructive hypertrophic cardiomyopathy. We are now including patients with obstructive cardiomyopathy. And previously we required a defibrillator. Now we don't require that defibrillator. So as you say, encouraging that the DSMB supports the dose escalation and the expansion in the eligibility criteria, so we can learn more about how we may see improvements in a broader population of patients with MYBPC3 related hypertrophic cardiomyopathy.
So they have increased the dosing allowed. They have changed the criteria to include both obstructive and non-obstructive patients. The ICD is not a requirement any longer. And we're adding in a biopsy at the beginning of the trial. So there will be a total of 3 biopsies through the trial. And for how long are you following these individuals who are being dosed?
So we follow patients for 5 years after the dose, which is a commitment for patients, but it's critical for science. As I said, we are anticipating that the effect will last through those 5 years, but we are using this trial to determine that. There are more visits at the beginning in the safety monitoring period, and for the years 2 through 5, it's basically annual visits, so not a lot of burden on the back end.
So let's talk about how success is measured here. It's not an improvement in NYHA class or a 6-minute walk test. It's different because it's gene therapy. And I know that the goals of any AAV9-driven therapy and any gene therapy is to deliver the package or the information that the heart needs to code properly, to deliver it to the heart so we meet the delivery demand or transduction. And we want to know that it didn't only get there, but it's doing something, and that we can measure through protein expression. Can you talk a little bit about those terms, why they're important, and what we're hoping to see?
We are measuring efficacy in this trial in patients over those 5 years with many measures. The early measurements will involve the science of it. Is the drug doing what it should do inside the cell, as you say? And with more patients and more time, we'll also be looking at clinical endpoints. Do patients feel better? Are they reporting better outcomes? Is their New York Heart Association class improved? We'll also be looking at imaging of the heart. So the thickness is improving and the function of the heart is improving. And we'll be looking at biomarkers of disease. I think many of your listeners know about NT-proBNP, for example. But the first things we'll learn, just even from the first few patients here, and we'll be talking about this in December, is about what's happening inside the cell in the heart after the treatment. Are the instructions getting to the cells, and are they being implemented by the cells? So that involves DNA, RNA, and protein. I like to think of it as furniture, where the proteins in your cell are like the furniture in your house. And so say, MyBPC3 is the kitchen table. So you got the parts from IKEA, but there were no instructions. So you just have parts lying around, right? But you
anybody who's put together an IKEA table knows that those parts all have a place, and if there's a piece left, it's not a problem.
The instructions are very precise. Yes, and one misspelling in the instructions, your table is not going to work. That's what we know from genetics. So you need the instructions. So what our drug TN-201 does is deliver a copy of those instructions. So that's the DNA. It needs to be adjusted a little. It's as if the instructions came in Swedish from IKEA. And so it's adjusted into English, translated essentially into RNA, which is what the cell can actually use to make the table. So that's the RNA. And so that the RNA is actually the instructions that should have come with your table in the first place. And then you use those to build the table with the parts that you have. But once you have those instructions, you can build, 5, 6, 7 tables and keep going. So we will be measuring those. First, the DNA, did that get to the cell? And so we can directly look at how many copies of DNA we're getting per cell in the heart. And we have, very clear targets. We know how much DNA we need to get to the heart for it to work in animal models, and we suspect it will be similar in humans. And then we can also measure the RNA level directly, and that's the instructions turning on, starting to be implemented by the cells. And then the protein, we measure those levels as well.
So this all sounds pretty fancy. How do you measure this stuff? Blood work? Is it biopsies? Is it tissue?
That's all from the heart biopsies. So we are performing heart biopsies as part of the trial. You've had a transplant, you've had biopsies. Correct me if I'm wrong. My sense from patients is that it sounds worse than it actually is in implantation. When you first hear about it, it seems like a much bigger deal than it is. But what do you think?
It is not a comfortable 10 minutes. But it's not an awful 10 minutes. I've never been sedated for a biopsy. Little lidocaine, an insertion into the neck. I carry on a conversation during my biopsies. I might drop a couple of F-bombs while they're putting it in because it hurts a little bit. But I'm from Jersey and that's how I cope. But otherwise, it's not that bad. And yes, there are risks associated with a biopsy. I have had over 20 heart biopsies. I kind of lost count, and this is 3. And obviously, you want a trained person who knows how to do a biopsy. So you're probably going to be meeting with a heart failure interventional cardiologist to have this done because they're the ones who do it. Typically, it's not horrible. It's not. It's a little unnerving, but we do big things. And we're tolerating HCM and the uncertainties and the fear of that. So in comparison, it's not that big of a deal.
We would not ask our participants to do this if it wasn't critical for understanding that our gene therapy is getting to the heart, turning on and making correct MyBPC3 protein. And the centers that are working with us on this study are all experts. They're all heart transplant centers with people that perform these heart biopsies all the time. So it's a routine for them.
Exactly. It's not routine for a patient, but it's definitely routine for the docs, and they know what they're doing in this space. I will just say that it's not 100% without risk. There could be, issues that come after a biopsy, but they're very, very, very tiny risks when done in high volume centers. So again, HCMA talking point 101, high volume matters. So the people who are involved in this study are high volume operators and they know what they're doing. So be reassured, because if you start reading up on biopsies, you might see some stuff that makes you nervous, but let's talk it out and talk to the providers who are actually doing the procedures before you decide, well, that's just not something that I want to do. I didn't want to do one either, but I had to, and it worked out really well.
Our patient participants are dedicated. They are committed not only to seeing whether the therapy will work for them, but often so that there'll be something available for their children and for other patients like them. So we have patients that are very eager to push the ball forward for science and for patients. But we strongly encourage them to have several discussions with the investigator, the HCM expert at the site conducting the trial, just to make sure and understand every part of the trial and the 5-year commitment that they're making. We encourage robust conversation.
So let's talk a little bit about participation. We know your goal has been increased from 15 to 24 trial participants. HCMA recently conducted a survey And in that survey, 6 individuals told us that they were enrolled in the program. I don't know whether they've been dosed or they're set to be dosed or they've been screened in or screened out. But there are a number of individuals who we know have either already been dosed or are in line to be dosed, but we need more to do that. Can you kind of walk through, obviously the clinical trial startup process for a patient is with that first engagement to say, I'm interested. Now, if somebody's interested and they want to talk to HCMA about it, you can call in, we can talk to you and connect you with the people at Tenaya who are doing the recruitment. Happy to do that. But if you're an independent individual and you really don't want an advocacy organization and you want to do it all yourself, here's the information that you need. We got to establish the connection and find you a study site. Can you talk about how many study sites are open right now, and can somebody travel into one, or is it strictly if you live in the neighborhood?
Yes, so we have 10 open in the United States now, and many sites will accept traveling patients, and we have done that. Certainly, and Tenaya supports all the travel expenses and reimburses for the hotel stay while you're there at the site. I can run through in alphabetical order by state. I'm not saying preferences here, but in California, we have UC San Diego and UC San Francisco. In Georgia, Emory. In Massachusetts, the Brigham and Women's Hospital. Minnesota, the Mayo Clinic. Ohio, we have 2 Cleveland Clinic and the University of Cincinnati. Oregon Health Sciences University. Pennsylvania, the University of Pennsylvania, and Houston Methodist in Texas. We've tried to make it relatively easy to travel to sites. We do welcome patients that aren't necessarily in those clinics at those centers already, having a conversation with the study teams at those sites and deciding whether the study is right for them.
The first step, I probably should have started with this one, you need to have genetic testing done. You need to know that you have myosin binding protein C, and that's kind of the starting point to then talk to the center about, is this right for me or not? What does the ideal patient participant profile, the triple P, what does the patient profile look like? Who do you want to see in these trials? Are there age limits? Are there disease status limits? Who should come in?
Yeah, we are looking for patients that have disease, not just the genetic variant in MYBPC3, but have hypertrophic cardiomyopathy, with thickened walls, who have symptoms, so New York Heart Association, class 2 or 3 to be specific, and other risk factors such as increased levels of NT-proBNP. With an adult study, you asked about age. So at this point, we are studying safety and efficacy in adults. And then once we've found the appropriate dose, we will initiate studies in children as well, including infants. So the patients listening to this that have MYBPC3 variants are almost certainly heterozygous, meaning they have one normal copy and one disease-causing copy. Unfortunately, if you're born with 2 disease-causing copies, you basically always need a heart transplant in the first year of life, if you can get it. So one of our missions and commitments is to get gene therapy to those infants as well when we have a safe and effective dose and agreement with the FDA.
I just want to go back on one of the inclusion criteria because we drop terms and patients don't always understand 100% what it means. So New York Heart Association is a gauging of how symptomatic a patient is. Doesn't work great in HCM all the time because we kind of fluctuate from one class to another. But I would look at your worst days or your average days, what your New York Heart Association class is, which is easily defined as how quickly do you become short of breath or symptomatic with exercise? Is it 1 flight of stairs? Is it 3 flights of stairs? Is it a hill? Is it walking across a flat floor? These are some indicators on what New York Heart Association class you will be. And anybody in an HCM family knows that we are good one day, bad the next. So you really kind of want to look at your bad days and your average days and talk about that with your physician. We all get a little holiday occasionally, and we love those good days, but we can't base our entire disease experience on the good days that we might get once in a while. We really need to look at our average and bad days because they will continue to increase in number if we don't get in front of them. So talk about your symptoms specifically. Another note here.
My sense is, and you know this better and can elaborate, is that many patients underestimate how limited they are. That's the way it is. And then after, say a myectomy or something like that, can actually realize how much, for example, they were not doing things without realizing they were avoiding.
This is the bane of our existence. We think we're normal and everybody feels what we feel, so we just have normalized it in our lives. And it's not normal to feel dizzy when you stand up. It's not normal to get short of breath just walking up a single flight of stairs. It's not normal to get chest pain after you've had a meal. Like, these things are not normal, but we do it all the time and we just accept it. You need to talk to your doctor about your symptoms. You're not complaining, you're documenting. It's okay to document what you're feeling because we now have so many tools to make you feel better. We need to know how you're not feeling good and how we can optimize your health. And to get into a trial like this one, you can't be perfect, because how do we measure success? You have to have some limiting factors so we can see that they've improved. And eventually, if this gets proven, there could be utility in the asymptomatic, but we're not there yet. And we're not going to be there for a long time. So slow roll, learn, get more data. Let's get down to being able to dose those infants who don't have an option. They're not going to get transplanted quickly. They're not going to live long lives. We, the adults, can do this for the children and then for all. And to everybody who has signed up to be in this particular trial, I literally thank you from the bottom of both of my hearts, the one in the box and the one in the chest, because maybe families won't have to be suffering. Maybe transplant won't be the destination for some of these people. And while I'm doing well with my transplant, not a journey that I'd want others to take if they didn't have to. So really encouraging signs here for participation. We know who you are, and we're going to be talking to you more specifically in the coming weeks about this. But if you have MYBPC3, if you're willing to do some travel or give the time, can you talk a little bit about the time commitment at the center? How long is somebody at the hospital when they're getting dosed? How long do they have to stay around the hospital? How much time do they need off of work?
There is a brief hospitalization. I'll get back to that. I do want to also shout out to our patient participants. You're the folks that make this work, and really allowing us to know whether we can directly fix the underlying cause of this disease. None of us could do this without you. So heroic in my mind, so thank you. We at Tenaya share your vision, Lisa, that ultimately this could be a preventative therapy for treating patients before they get symptoms or before there's any damage to their heart. But we want to get there safely, as you say. I will caveat, you say we're a long way off, but long is very relative, and this is a disease that has affected generations in your family and your listeners' families, and now we're talking months and a few years, not generations and everything. So the time really is now. I know on the physician side, geneticists, genetic cardiologists have been waiting for the tools to do gene therapies correctly for many, many years to help the families that they treat. And really, this is the time. We are turning a corner here. Okay, so I promised to talk about the safety monitoring at the beginning of the trial. So there is a hospitalization for the first 8 days. It's boring. That's what we hear, that it's like, bring entertainment or bring your work, because really what we're doing is measuring blood tests every morning to monitor the immune reaction. And the vision is, when we understand how the immune system is reacting to the TN201, and in particular the time course for that, we'll be able to simplify the monitoring and potentially even not require the hospitalization in the future once we know the time course and what's changing. So we do a large panel of immune monitoring blood tests over time during that period. And then after that, there are frequent visits for blood tests for monitoring. We do know that gene therapies like ours can irritate the liver. The immune system's reaction irritates the liver. So we follow that weekly for a while. Patients can often do that at a local laboratory. They don't need to go to the study site. And the duration of that depends on individual patients. Everyone's immune system is different. And so we're carefully following that. And once that period has passed for that patient, then the monitoring decreases substantially. And so, by the end of the first year, it's visits, every 2 to 3 months. And again, in the follow-up years, it's every year.
So let's talk about the immune response. I am a person who is immunocompromised through transplant medication, and I've been on this for almost 8 years now, surprising, but I'm still here and I'm on immunosuppression. Talk a little bit about the trial and immunosuppression and how long it's necessary for at this time.
Yeah, and you are not alone. You have large numbers of fellow patients that have had successful transplants and are on lifelong immunosuppression therapy to keep the immune system from reacting to and rejecting your new heart. The gene therapy, the mechanism that we use to deliver the instructions is foreign to the body. The body will want to reject it, just like rejecting a heart. Much smaller, much less complicated than a whole heart. Many fewer things for the immune system to recognize, and it's temporary. The delivery mechanism is like the box, the FedEx box that the instructions are in. It gets recycled. It literally gets broken down and remade into other parts after weeks to a few months. So we do use immunosuppression. It is similar to immunosuppression in patients who've had a heart transplant, but much shorter period of time, measured in weeks to months.
And what other medical management do they need while they're in the hospital? They're getting a virus. Do they get sick?
Some people feel a little bit of GI upset, nausea about it, but no, not really. As I say, that experience we hear about, it's more about
boredom. Not a bad side effect if you can keep
now
it
that,
to that.
You've had prednisone, the mainstay immunosuppression we use, does have its own side effects that folks should talk with their doctors about before transplant or before temporary use of it. And what was your experience with prednisone?
Okay, so story time with Lisa. So the Christmas before my transplant, I bought my husband a really nice bottle of scotch. I mean, it was really nice. It was expensive. And I signed it from crazy prednisone girl to calm, sorry, because I thought I was going to be, you can get really intense when you're on prednisone. I became obnoxiously happy on prednisone. Sarcasm was my language, and everything was punctuated with a joke. That was my side effect to prednisone. So it wasn't so bad. So I took the bottle of scotch back and I said, you don't get to keep this, but he kept it anyway. So my prednisone experience was more euphoric and more amped up. Believe it or not, I could get more amped up than I normally am. So it was intense, and I didn't sleep well. And then they took it away and I was fine. And I did gain a little bit of weight on it. So it wasn't for a long period of time, and it went away, and I got my sleep back, and we're fine today. And I've done little prednisone adventures post-transplant for backache, hip ache, inflammation and things on that nature. And it just keeps you up and awake. So if you got work to do, great time to get it done because you're going to be awake. That's prednisone.
Right. And again, it's temporary, and we decrease the dose very quickly as we monitor. Again, that's what I said. We monitor the liver, and as appropriate, we decrease the dose of prednisone rapidly. That's why we're testing so frequently, is so that we can decrease the dose rapidly.
So we talked about the dosing. We talked about the immunosuppression. We talked about timelines for surveillance. But there's some other tests that are going to be done. They're kind of basic for HCM. We've got the biopsies, which we talked about, echocardiograms, which we're all very used to doing. CPET, you want to talk about what you're interested in seeing in CPET?
Cardiopulmonary exercise testing, or CPET, is a really fancy way to measure heart function. But that's essentially what we're doing is measuring how much blood can your heart pump and how much exercise does that support. And when I say how much blood does the heart pump, it's specifically the oxygen capacity that I'm talking about. So patients with heart diseases often are limited in what they can do. And so as you push the heart higher, it goes, the body goes anaerobic more quickly because there's not enough oxygen getting to the cells. So anaerobic means the cells start making energy without using oxygen. That's when you start feeling your muscles burning and that sort of thing. So on this test, it slowly increases the amount of effort up to that point where you feel your muscles burning and you're breathing very heavily. And then it measures the amount of work that you're able to do. In other words, that's kind of speed that you can travel uphill at that maximum effort. But we do measure the effort along the way as well, because we recognize that most patients aren't getting to that level of exertion in their daily lives. And we also want to understand how this drug is improving exercise capacity in daily life. So we are looking to improve both maximum exercise capacity and comfort with daily activities of daily living and lower impact exercise, so that patients can do more. So that's
we're also using
we're looking for.
Also using Kansas City Living with Heart Failure questionnaire and a 6-minute walk test. If you've been in any clinical trials in HCM, you took KCCQ, shout out to John Spurtis, and then the 6-minute walk test, which I think is probably one of the weakest prognosticators in HCM, but it's a good endpoint to see if people do a little bit better on that 6-minute walk test over time. We know HCM patients can walk on flat surfaces for a long time, so it's a good measure for a trial, but maybe not the greatest indicator in real life. This is a first-in-human clinical trial, and there's been a handful of people who've already stepped forward, and we'll learn more about them in December. We're kind of hoping maybe you'll come back and talk to us again in December about that data. So here is the public invitation to do so. So can you talk a little bit about first-in-human trials? And they're not for everybody, but who should consider this?
Patients who are symptomatic from their MYBPC3 heart disease, who are looking for new options, and maybe you've exhausted all your options, or maybe just the current options don't seem right to you. And part of your personal mission is to help others. Many of our patients have children that they know are carrying the gene, and they are really interested to make new options available for their children, and for just other patients as well in this generation and future generations. And I like the way you put it earlier, including those infants. Most of you won't know families affected, but they've had infants die from MYBPC3 cardiomyopathy. But they're out there, and they could all use our help.
Absolutely. When we were doing our planning for this call, you used a phrase that I wrote down and I really liked it. We are currently the greatest generation in HCM discovery. As a collective ecosystem, we are doing work that I only dreamed about 30 years ago. And it's not just the patients who are the greatest generation when they put themselves forward to participate in a clinical trial, whether it's for a myosin inhibitor, whether it's for septal reduction therapy evaluation, or it's genetic therapy. We're doing the work so that the people that come behind us can benefit from what we've learned together. And it is truly an extraordinary time. It is it's exhausting for some of us because there's a lot of work to be done, but it's so exciting to be able to utter the words, "Is a cure possible?" I didn't use the C word for a long time, and I'm just getting comfortable with it because it's a big word. It's a big concept. Improvement of quality of life, great. Extension of life, fabulous. Eradication of disease process, exciting. And it's a little hard to keep the enthusiasm under control. We have more data. So everybody's kind of watching and waiting to see where this is going to go. How long is this process going to take, do you think, to get to the answer of this is how we're going to use this tool? or no, this tool's not going to work.
There are many ways that patients can help and be part of the greatest generation of patients, really storming the trials and participating in trials, moving the science forward is a key way. Participating in trials isn't for all patients, but there's many other ways that you can help. Motivating physicians to participate in trials is part of it. Patient advocacy organizations are often partnering with physician groups to really emphasize that what unmet needs are still there and what patients are really hoping for next, so that the treatment community doesn't get complacent with what's available now. And we have participants with that kind of level of enthusiasm that they're helping push forward. But joining patient advocacy organization like HCMA, also critical, influencing the government as well, like the FDA and the patient-focused development conferences, so that the FDA really knows what patients are looking for next. Contributing at biotechnology companies. So we have a patient advisory council. We've had it for years. I have learned a lot from those patients. Thank you if you are on the call. It helps us think about the best design for our trials, what we should measure most, how we should communicate what we're measuring in informed consent forms, what wording resonates the most accurately for patients and everything. So that is another key way that we find very valuable for patients to contribute.
We have 24 slots in a trial. Some of them are already filled. We'll get a number hopefully in the next couple of months. So there's space for those who are interested. Ladies and gentlemen, this is the time. If you are interested in participating in a clinical trial with a myosin binding protein C mutation and you are symptomatic, this is your moment. You need to have conversations. You need to talk to your care team. You need to talk to the clinical trial specialist. You need to talk to a PI at one of the locations about what this all means. We're giving you the 30,000-foot view. We're telling you what's possible. We need to learn more. I can't say that being in a clinical trial is 100% lovely, wonderful, and fun, but it's meaningful work. Yes, you might be bored for 8 days in a hospital. That's the least of the problems. You may wonder, what is this doing? Am I okay? You're going to have people to talk to. You're going to have to communicate with. You're not doing this alone. And we, the community, applaud you if you want to step forward and do this. And if it's not right for you, no harm, no foul. Move on to the next thing. I've got other clinical trials coming up that might be more to your liking. So there's going to be a lot of trial opportunities in the next 5 to 6 months that are going to become available. But this is the one for gene therapy now. This is it. There was another company in this space. They've backed out. Don't read anything into that. Their company's going in a different direction. It's not that there was a problem with this. It's there was more opportunity elsewhere. That's the way we are viewing their move. So thank you, BioMarin. Moving on, maybe you'll come back. But Tenaya is here. I visited your plant. I visited your office. I've seen the production facility. I know that you guys are scaling up to produce, not only test. So I look at those signs as positive signaling. If you weren't seeing positive signaling, there would be no need to build the infrastructure that you've built, which is quite impressive. So I just want those who are on the fence to stop and think, if you guys are putting all this effort into building up a development process, a production facility, the DSMB has made some moves to the positive. I read these all as positive science. We'll get more data in December. What else do we need to know, Whit?
The time is now. This is the generation, and this is the year to really participate. You asked about what to expect in the future. I do think the Danon program for Danon hypertrophic cardiomyopathy provides a roadmap and an example of the FDA also helping bring new therapies to patients and the FDA's interest in addressing the underlying cause of disease. The Danon program had a very similar design for their first-in-human study. a small number of patients, found the right dose, and then they went directly into the pivotal study. So the one that will allow the FDA to make a decision about whether to approve the gene therapy or not. And then that's already fully enrolled. So, this can happen in a few years' time. And it does, though, depend on patient participation. And the more people to participate, the faster we'll be able to do this.
We have a question coming from France. Fabienne says, thank you for the work. If it works, will you need to conduct trials for each gene in the future? Or once it's proven to work, the formulary can be adjusted to deploy across the entire panel? That is a very thought-provoking question, Fabienne, and I will drop that onto Whit.
Thank you for pointing that out, Fabienne. That is a key point, that this therapy that we're developing is specific for the MYBPC3 gene. So the drug delivers a working copy of that gene and only that gene. So myosin heavy chain, the second most common cause of hypertrophic cardiomyopathy, will require a whole new approach. We're kind of circling back to the beginning here. In the case of the myosin heavy chain, MYH7, that gene has a misspelling that creates protein that is having the wrong effect. So a bad protein, we need to either correct it through editing, so fix that spelling mistake, or block it to turn it off. So that's a different approach. It will require a different drug that will require its own set of trials, both a first in human and drug dose finding study and its own pivotal trial. We are working on that. Editing has promise for the future. It's more complicated to deliver to the heart, so it's not moving forward as quickly as the MYBPC3 program, but we are definitely optimistic that in the future we will have a portfolio of different drugs and different gene therapy strategies to personalize the therapy and address the specific genetic mutation with the appropriate gene therapy.
Fantastic. Fabienne, I hope that answered your question. Any other questions? We're going to be wrapping in a couple of minutes, so now would be the time to post a question. We've gone through an awful lot of information on a really heady topic today, and I've enjoyed the conversation, but there's so much more to talk about here, right? It's an evolving space. The work that the FDA is doing, they've created a new department specially focused on genetic therapies because they see what's coming. If our federal government is amping up its regulatory agencies to respond, we know that this is a path for the future. We know that other disease states are looking at genetic corrections or genetic therapies as well. And this is a really exciting time. There are a lot of questions that we can go into later about cost and how we're going to deliver this and what does commercializing something like this look like, and how do we get people to get the therapy safely. This is a starting point that opens up all those other doors. And here at the HCMA, we have learned from other drugs that we've been involved with trials. Some make it, some don't, some go to market. You see commercials on TV now. We know that while we're in this discovery stage, we need to be thinking about the regulatory and the access issues early in process. So we're working behind the scenes here to draw out those playbooks and figure out what we're going to have to do later. But before we can deliver hope and therapies to people, we need to prove concept with strong science. And we need trial participants. Is that rounding it up nicely?
More than nicely. Very, very well said.
Yeah, thank you from Fabienne. You're welcome, and we are going to wrap up here in a moment. If this conversation has enticed you to think a little bit more about participating in a trial, I'm going to ask you a favor. I'm not asking you to sign up for a trial. I'm asking you to have a conversation with your physician, with a trial coordinator, or a PI, a principal investigator at one of the sites. Talk to them about your individual options. Talk to them about your individual HCM progression, what's happening with you, what's happened in your family. Is this something that is right for you and your family at this time? This is an incredibly personal decision. And for those who decide it's not for them, I respect it. For those who think it might be right for me, talk it out. Find out if it is right or wrong for you, and then make your decision. But let this be the spark that made you think, yeah, maybe I'm interested. and continue those conversations. I can't say how important it is any stronger than what we've done in the last hour. Without trial participants, we never get to the finish line. And that means somebody's going to accept some risk. And for that, I thank you for being brave. To those who have already dosed, I thank you for being brave and for thinking about others and yourself. It's okay to think about yourself in this. It's okay to want to feel better. That's not a bad thing. We deserve it. We deserve a good quality of life. And it is now more available to us than it's ever been. And the hope of life without HCM is, without a transplant, is actually actionable for some people now. Boom. Crazy ideas. Whit, final thoughts.
Again, very well said. Thank you so much for inviting me. And I do look forward to sharing more news in the future. And thank you, everyone, for listening and for participating in the HCMA and in general, working to understand your disease better and working to think through how the patients like yourselves can join together to create new options.
Parting messages as we go. I want to welcome Artie and Maria to the HCMA team. They are our new intake coordinators, and you're going to hear from them soon. They're training up in the room behind me right now, got locked in a conference room for a month or so. So be nice to Artie and Maria. And another big announcement that has not been said yet, we have another new contractor, Juliana Krem, who resides in São Paulo, Brazil. We'll be working with our HCMA International program along with Michelle, and we welcome Juliana to our team as well. We are growing, and we are actually have a job posted right now for communications manager. So if you are in the North Jersey area and can work out of Denville, New Jersey, that job is now posted and we are accepting applications. So come join us, grow, and see what amazing things can happen here at the HCMA. We're not only a great organization to serve the community, but we're actually a pretty damn good employer too. And a large part of our staff actually are affected by HCM. Bet you didn't know that factoid. Next week, the summit in Boston. Whit, you and I will be in a room together next week. And we also have the HCMA annual patient meeting Sunday, October 27th in Boston. If you haven't signed up, please do. And if you are attending the summit, remember to sign up for Saturday night at the summit with the HCMA. We are limited to 150 people and we are close to sold out right now. So please sign up if you want to join us. We'd love to have you spend some time with us. There's Artie saying hello. Say hi to Artie. And we are excited to have you on the team, sweetie. All right, Whit, thank you. To all of our friends at Tenaya, Faraz, Wendy, the entire team, thank you for everything that you're doing for patients. Continue the hard work. It's worth it. We appreciate you every day, even if we don't get to say it. Thank you all for joining and have a great day.
The Hypertrophic Cardiomyopathy Association thanks the following sponsors for today's podcast: Cytokinetics, Bristol Myers Squibb, BioMarin, Tenaya Therapeutics, Viz AI, and Alnylam.